Hello, my name is Abby Romano, and today we'll examine mammary alveoli, the secretory units of the mammary gland, through a lens of anatomy, physiology, and emerging research. We'll connect structural biology to functional outcomes and highlight how cutting-edge models are reshaping our understanding of lactation. So, alveoli are the smallest functional units of the mammary gland, yet they dictate milk yield and composition. Each alveolus is a microenvironment where epithelial cells synthesize milk under hormonal and metabolic control. Recent breakthroughs like ruminant mammary organoids allow us to replicate alveolar architecture in vitro, enabling mechanistic studies of lactation and diseases. This matters because traditional rodent models don't fully capture ruminant-specific physiology, which is crucial for dairy science. So, the gland's architecture follows a hierarchical pattern: lobes to lobules to alveoli. Each alveolus is a hollow sphere lined by secretory epithelium and surrounded by myoepithelial cells. Histologically, alveoli differ from ducts by their lumen size and secretory specialization. In buffalo, about 95% of milk resides in alveoli, whereas dairy cows store more in cisterns. This anatomical difference influences milking protocols and oxytocin response. So, for cellular architecture, two epithelial layers mainly define alveolar function: the luminal cells, which exhibit polarity and synthesize milk components, and basal myoepithelial cells, which contract during milk ejection. Tight junction proteins maintain barrier integrity. Stromal fibroblasts and extracellular matrix components provide structural cues and signaling that regulate alveolar development. So, alveologenesis is exactly what it sounds like, the development of alveolar. So, alveolar development accelerates during late gestation under endocrine control. Estrogen and progesterone drive ductal elongation and lobulo-alveolar expansion, while prolactin and growth factors like EGF and FGF2 induce differentiation. Species variation is notable. Pigs exhibit rapid alveolar proliferation in the final trimester, whereas goats and cows show more gradual expansion. So you can see in these figures below that in the embryogenesis phase, you have some structure there, but definitely no alveoli. The alveoli are those purple masses that you see are the groups of alveoli, and those we begin to see in gestation. So, the functional biology of these alveoli, milk synthesis involves coordinated metabolic pathways. So, glucose uptake fuels lactose synthesis. Fatty acids are esterified into triglycerides, and casein and whey proteins are synthesized in the rough ER, packaged in Golgi vesicles, and secreted via exocytosis. Tight junction closure is essential for maintaining osmotic gradients and preventing cell-to-cell leakage. These processes demand high ATP turnover, explaining the nutritional sensitivity of lactation. So that is especially why in high-producing dairy cattle, their nutrition is so vital to their milk production. To talk about milk ejection, oxytocin, released from the posterior pituitary, binds to receptors on myoepithelial cells, triggering intracellular calcium influx and actin-myosin contraction. This compresses the alveoli, forcing milk into ducts. So think of it as kind of a balloon, and the myoepithelial cells are squeezing it, and the milk is exiting out of the alveoli. The reflex is neuroendocrine. Tactile stimulation of the teat activates hypothalamic neurons. So stress-induced cortisol can antagonize oxytocin signaling, which reduces milk flow. And this is a key management concern in dairy systems. So, immune and barrier functions. Alveoli host innate immune defenses. Macrophages and neutrophils patrol the lumen, while epithelial cells secrete antimicrobial peptides. Tight junctions prevent pathogen invasion, but disruption during mastitis allows bacterial colonization. Inflammation alters gene expression, upregulating the cytokines and downregulating milk protein synthesis, leading to yield losses, and this is detrimental in the dairy industry. That's why understanding alveolar immunity informs mastitis prevention strategies and therapeutic interventions. So, the mammary alveoli are actually very, very impressive. They have the ability to kind of remodel and regrow themselves after going through involution. So they undergo dynamic remodeling across the reproductive cycle. So during involution, apoptosis of luminal cells and extracellular matrix degradation occur. Some changes are reversible, but prenatal threats like heat stress can permanently alter alveolar size and connective tissue composition, thus reducing lifetime milk yield. This underscores the importance of managing environmental stress during gestation. So alveolar dysfunction manifests in mastitis. It can also manifest in neoplasia and metabolic disorders. Mastitis disrupts tight junctions and induces inflammatory cascades, while neoplastic transformation involves dysregulated proliferation and loss of polarity. So that dysregulated proliferation just means growing at an unregulated rate. So comparative studies reveal species-specific susceptibilities to this. Buffalo's thicker teat sphincter reduces infection risk, but complicates machine milking. These nuances guide species-tailored management and therapeutic approaches. So it's really cool. There's a lot of emerging technology surrounding the mammary gland and the dairy technology. Specifically, 3D organoid cultures that replicate alveolar architecture enable long-term lactation studies and genetic manipulation. These systems respond to lactogenic hormones and produce authentic milk proteins, which offers platforms for mastitis research and cell-based dairy applications. CRISPR and single-cell transcriptomics, which is the study of all RNA molecules in a cell, are mapping lineage hierarchies and uncovering regulatory networks. Future directions include organ-on-chip models and bioprinting, which could revolutionize both research and commercial milk synthesis.